Welcome back. So I'm finally getting around to doing the mechanism and physiology of catalase. So catalase is this enzyme right here. And before we get into the heavy-duty mechanics of this enzyme and some details about it, let's talk about the net reaction. Okay, so number one, it's going to take two molecules of hydrogen peroxide to do this reaction in one full cycle. So this molecule right here that I've drawn, this is called hydrogen peroxide. Okay, we'll eventually have a whole video on hydrogen peroxide and all the nasty things it can do. For example, it could do a homolytic bond cleavage in which it would basically generate two hydroxyl radicals, or it can undergo things like Fenton reactions if it comes in contact with free iron. And Fenton reactions also produce hydroxyl radicals. And overall, this molecule does damage to proteins, it does damage to membrane lipids, all sorts of nasty things you don't want. Okay, so catalase is going to be the enzyme that gets rid of it. Okay, and we'll look at the mechanism of how that occurs in just a few minutes. Okay. Just some other things about catalase is it's expressed both inside the cell and outside the cell. Okay, so it's expressed, number one, in really high concentrations in an organelle that's known as the peroxisome. Okay, a lot of reactions that produce hydrogen peroxide are localized to the peroxisome. Okay, so there's also a cytosolic catalase, just in case you were to need that. But also peroxisomes contain a high concentration of catalase because there are a lot of enzymes that produce hydrogen peroxide, and so they're localized to the peroxisome to theoretically keep most of the hydrogen peroxide species under control inside one organelle where you have high concentrations of this enzyme. Okay, but it also exists in the cytosol because sometimes you can get hydrogen peroxide in there. And there's also an extracellular extracellular catalase that can destroy extracellular hydrogen peroxide. So what would be the sources of extracellular hydrogen peroxide? Well, if you're eating inflammatory foods like dairy and gluten, and to some extent even things like soy, you're going to be getting things like respiratory burst, ultimately due to things like antibody activation in the complement system. So there's some application there, but in any case, you can have extracellular hydrogen peroxide in the blood or something, so you have to have a way to deal with it out there, and extracellular catalase is what does that. Okay, so a little bit about the structure of the enzyme. Number one, this enzyme is going to have a heme B coenzyme. Okay, and you'll notice that in the center of heme B, of course, we have this iron that starts out the mechanism, all mechanisms in this case, in the ferric state. Whenever I say ferric iron, I mean iron that's in the 3+ oxidation state. You could also have ferrous iron, which would be the case in something like hemoglobin, myoglobin, neuroglobin, cytoglobin, things like that. But in general, with enzymes, it starts out in the 3+ state. So whenever I show iron down here like this, so this is iron, you're to assume that the porphyrin ring of the heme system is around it, okay? I just obviously don't show that in every mechanistic step due to the complexity of the porphyrin ring, but you're to assume that it's there, okay? So that's the coenzyme or cofactor that's used in the reaction of catalase, okay? Also of note in the active site, there is a critical histidine residue that's going to act as a Bronsted-Lowry acid-base donor and acceptor. Okay, so that's the Bronsted-Lowry general acid-base catalyzer. One other thing that's important about this reaction is it's one of the few reactions in biochemistry that's classified as a disproportionation. So disproportionations are often things that you have in the inorganic chemistry perspective, and in this case, this is an inorganic reaction. Yes, of course, you have the histidine, and of course the porphyrin ring has carbons, but in terms of things that are doing direct catalysis, you have the nitrogen of the histidine, and then you just have hydrogens and oxygen atoms. So this really is an inorganic mechanism, and of course the iron would also be classified as inorganic. Okay, so it's mostly inorganic. You can even consider it all inorganic. And disproportionation reactions are really common there. So to give sort of a gross generalization of what a disproportionation reaction is, it's when you have something, some molecule, and you could consider either the semi-reduced or semi-oxidized oxidation state, And basically, you kind of split the molecule apart, or you'll have two molecules to start, and your final products go from semi-reduced to fully oxidized or fully reduced. Okay, so for example, if you look at the structure of hydrogen peroxide, which is shown here, okay, this oxidation state for the oxygen here is sort of intermediate, okay? It's in the intermediate state. So basically, you have the oxygen hogging electrons from the hydrogen, but what's going to happen in this reaction sequence is the molecular oxygen, part of it's going to get oxidized into molecular oxygen, as shown right here, and then the other parts of the oxygens are going to be totally reduced to water. So let me do that in blue. So it's going to be totally reduced to water. So the water represents the totally reduced form, and then the totally oxidized form is the molecular oxygen. So this is the oxidized form. So we're going from something that's semi-reduced / semi-oxidized, whatever you want to call it, to products that are either totally oxidized or totally reduced. Okay, so the key is that the products are more oxidized and more reduced depending on which product you're talking about, and that's what classifies a disproportionation reaction. Okay, and the way we're going to transfer the electrons is going to be ultimately dealt with through the heme ion. Okay, so let's actually look at the mechanism of how this occurs. So the first step, which I'll do the mechanistic steps in green, the histidine, critical histidine residue in the active site, is going to deprotonate the hydrogen peroxide, causing an electrostatic interaction to form between the anionic peroxide species and the iron 3+. So all electrostatic interactions in this mechanism are shown in yellow. Okay? So you have to imagine on this iron, there's of course, some orbital or whatnot that has electrons in it, 2 electrons we'll say. And so what's going to happen is sort of at the same time that these electrons come and attack this atom of oxygen, the effective hydroxide is going to leave and re-abstract the proton from the critical histidine residue in the active site. So that gives us our first product, which is water. This is our first product, water. Okay, and that of course regenerates our deprotonated critical histidine residue in the active site. Okay, and what that generates is an oxide now that is in a coordinate covalent bond to the iron, which you could effectively view as being in the 5+ oxidation state. Okay, and now at this point what's going to happen is we're going to do some radical mechanisms. So part of the genius of having heme in this mechanism is what heme allows mechanisms to do is operate by radical mechanisms. Okay, the electronics of the heme ring and the axial ligand allow this to take place. Okay, so the first step is you're basically going to get, number one, a homolytic bond cleavage in this coordinate covalent bond. So one of these electrons is going to essentially abstract this hydrogen right here, okay, forcing a lone electron onto this atom of oxygen, and then the other electron here is going to be forced onto the iron in the 5+ state. And what that does is it generates iron in the 4+ state with a chelated hydroxide. And then of course we end up with this basically protonated superoxide. Okay, the last step of the mechanism is going to be another homolytic bond cleavage where one of these electrons ends up back on the iron 4+ to make iron in the 3+ state to regenerate the mechanism, and then the last one of these electrons comes here and abstracts this proton, and then what you end up generating there is you end up generating molecular oxygen. Okay, so of course, as you can probably guess, this right here, this represents the molecular oxygen, and then the last product is going to be water. So we also regenerate the iron in the 3+ state, which is the ferric state of iron. So basically, we go from 2 molecules of hydrogen peroxide, which are very bad for both outside the cell and inside the cell, and we convert them to products that are basically harmless, which are 2 H2O molecules, 2 waters, and then we also get molecular oxygen. Okay? So by doing this reaction, we basically disallow hydrogen peroxide from exerting damage on cells or inside cells, basically. So hopefully this video gave you a little bit of intuition on catalase. In the next video, we're actually going to look at some of the properties of hydrogen peroxide and what kind of damages it can do to cells given various reactions. See you in the next video.